ribulose bisphosphate, that is RUBP. Ribulose, it's a 5 carbon keto sugar ribulose bisphosphate 2 phosphate, so it is RUBP. Okay, this is the primary acceptor of CO2 in C3 cycle or C3 pathway given by the Kelvin scientist. So this Kelvin pathway occurs in all photosynthetic plants. All photosynthetic plants. It does not matter whether they have C3 or C4 pathways. Okay. Why plants are C3 and C4? Because based on the first stable product of CO2 fixation. See, CO2 fixation occurs in both C3 and C4 plants. But based on the CO2 fixation product, if the acid is oxaloacetic acid or OAA, it's written in here. If it is oxaloacetic acid, OAA, that is 4 carbon compound or 4 carbon acid, then this pathway is C4 pathway. Then the pathway is C4 pathway. And C3 pathway, if the first CO2 fixation product is a C3 acid, that is PGA, phosphoglyceric acid, that is PGA. Okay, so two main types, those plants. So plants are differentiated into C3 and C4 based on the first product CO2 fixation. So if the acid is, that is first stable product is PGA, then the pathway is C3 pathway. Okay, plant showing the C3 pathway. And if the first product was C4 acid, that is oxaloacetic acid, then the pathway is C4 pathway or plant showing the C4 pathway. So this was it. We have seen the primary acceptor of CO2. What causes this? The primary acceptor, that is a fixation of CO2, is a molecule, that is ribulose, which is a 5 carbon keto sugar bisphosphate, because it has 2 phosphate. So 5 carbon keto sugar ribulose bisphosphate, RUBP. So in short form, it is ribulose bisphosphate, RUBP, right? Remember that again, C3 cycle is given by the Kelvin scientist, right? And this Kelvin pathway occurs in all photosynthetic plants. It does not matter whether they have C3 or C4 pathways, right? It does not matter whether they have C3 or C4 pathways. Now, so Kelvin cycle. Now we see the Kelvin cycle. Kelvin cycle, that is C3 cycle. The first stable fixation CO2 fixation product is PGA, that is the C3 pathway. Okay, also known as Kelvin cycle. So Kelvin cycle has 3 stages. Number 1, carboxylation, where CO2 fixation occurs, okay, with RUBP, and formation of 2 molecules of 3-PGA, 3-phosphoglyceric acid. And the full form of this RUBP enzyme is RUBISCO, Rubisco, because it can fix oxygen in photorespiration and CO2 in generally C4 and C3 as we are seeing. Okay, so Rubisco can fix CO2 and O2. You have to remember this now. Next thing, next thing is the reduction. So the second step is reduction and the third step is the regeneration. Okay, now in the first step we have seen the carboxylation. Carboxylation means carbon fixation, that is CO2 combines with the RUBP 1,5 bisphosphate. It's a name of that enzyme, ribulose 1,5 bisphosphate, that is RUBP. Now see the figure 11.8. It's clearly given that the Kelvin cycle proceeds in three stages. The first one is the carboxylation, during which CO2 combines with RUBP, that is ribulose 1,5 bisphosphate. Okay. Now in the second process is the reduction, during which carbohydrate is formed at the expense of the photochemically made ATP and NADPH. Remember, now ATP and NADPH are formed before all these C3 and C4. It's a currency right now. Now plants using this currency, right, that is ATP and NADPH. So reduction is the stage where carbohydrate is formed. What carbohydrate? It's glucose, sucrose, starch. Okay, in the expense of the photochemically made. Photochemically made means through the light reaction. Okay, non-cyclic light reaction before this Kelvin cycle, right? So now photochemically made ATP and NADPH is used. Now the third step is the regeneration. Now third step is the regeneration. So during which the CO2 acceptor ribulose 1,5 bisphosphate is formed again so that the cycle continues. Now the thing is, in the first step you see ribulose 1,5 bisphosphate is fixing this process, and in the third step there is a regeneration of this ribulose 1,5 bisphosphate. Okay, so the cycle kept continues, right? So in the first step it's a carboxylation, that is the use of ribulose 1,5 bisphosphate. In the third step is the regeneration of that molecule, so the cycle continues. Okay, and in the okay, so in the second step you have seen there is a expense of the ATP and NADPH. The same in the regeneration is the 1 ATP for phosphorylation to form the RUBP, because RUBP contains phosphorus. ATP has also phosphorus, so ATP is used to regenerate this RUBP, right? Now 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 you see, now talking about the conclusion of the Kelvin cycle, you have to remember that in the reduction process, in the reduction stage, 3 molecules of ATP and 2 molecules of NADPH are required. Okay, okay, so 3 molecules of ATP and 2 molecules of NADPH are required for every CO2 molecule fixing in the carboxylation stage of Kelvin cycle. Now we want a glucose. Now glucose has C6H12O6 molecule. So these are the carbon molecules 6 carbon molecules. So we want the fixation of, because CO2 has C carbon, so we want six CO2 molecules, that requires a 6 turns of the cycle, because we want the glucose C6H12O6, 6 carbons we want. Okay, so now the total ATP and NADPH becomes, okay, for every CO2 molecule entering the Kelvin cycle, 3 molecules of ATP and 2 molecules of NADPH. So 6 into 3 is 18 ATP, and 6 into 2 is 12 NADPH, right? So 6 CO2 gives 1 glucose molecule, and 18 ATP gives 18 ADP, and 12 NADPH gives 12 ADP and ADP. So this was the Kelvin cycle that process in 3 steps, that is carboxylation, reduction and regeneration. Also in given in here, that is utilization of 2 molecules of ATP for phosphorylation and 2 molecules of NADPH for reduction per CO2 molecule fixed. Okay, so reduction involves utilization of 2 molecules of ATP for phosphorylation and 2 molecules of NADPH for reduction per CO2 molecule fixed, right? And in regeneration, 1 ATP molecule is needed. So total 3 ATP molecules needed in the Kelvin cycle, and 2 molecules of NADPH needed in the Kelvin cycle. And we want glucose, that is 6,6H12O6. So it has 6 carbon atom. So we want 6 CO2 fixation. So 6 into 3 is 18 ATP, and 6 into 2 is 12 NADPH.